The ancient genome analysis was conducted by geneticists at Trinity University to discover how goats were first domesticated. To investigate patterns and processes of goat domestication, they extracted DNA from the petrious bone located in the inner ear of goat skeletons found in the Fertile Crescent region of Southwest Asia. They sequenced the DNA, which gives a detailed picture as to what the earliest herd field populations looked like. In order to extract the DNA, they must be very careful about contamination. They take them into a specialized ancient DNA lab where they are cleaned and UV'd to remove any contaminants. From this bone, they were able to find the pattern of domestication was mosaic, meaning there were two different genotypes from a single fertilized egg. They also found the oldest described catalog of genes that have been selected by humans. From their research, they found the genes involved coat color and pigmentation, providing evidence that domestication was repeated and happened in different places by people across the world. Their evidence allows them to continue research to compare what is similar and different to ancient and modern goats of today's world.
Specifically related to the Trinity research, we are going to go more in depth on how goats are domesticated today through mitochondrial inheritance. Genes are specific DNA arrangements on chromosomes which are found in the nucleus of each cell. Within these cells are going to be important genes on the DNA found in another compartment of each cell called the mitochondria. Mitochondria and the DNA inside it are passed from one generation to the next throughout the mother's egg cells. Considering most of the mitochondrial DNA comes from the mother's egg and only a few come from a father's sperm cells, this inheritance pattern is commonly referred as maternal inheritance. In this example here, we have an affected mother of a specific gene mutation and an unaffected father. The mitochondrial mutation is inherited from a mother whose own egg cell can contain both working and faulty copies of this mitochondrial gene.
In a man-made migration event, producers would occasionally recruit wild types for their herd. This led to more variation in the genotype that evolved over time into the different goat populations seen today.
Trinity decided to study the history of goats' domestication through the mitochondrial DNA. Because of this, these mutations and specific traits such as coat colors get passed down from generation to the next.
Mitochondrial genome determined that over 8,000 years ago, ranch producers viewed goat color as the most valuable product, and only later did they find the importance of meat, milk, and reproduction management.
Goat genetics rocks!